This is BIO367 Lecture 8, Part 2. In this part of the lecture, we're going to talk about global control mechanisms, which is a topic that still falls under the larger category of pre-transcriptional gene regulation. A pattern of regulation often seen in bacteria and archaea is something called a global control system. Global control systems are regulatory systems, that respond to an environmental cue by impacting the expression of many seemingly unrelated genes. Probably the best studied example of a global control system in microbes, perhaps in all organisms, is the control system known as catabolite repression. This is a control system that affects the expression of the operon. It's also sometimes referred to as glucose repression. And we're going to review, what I hope is review, that particular system next to introduce you to some of the concepts important in global control systems. To understand catabolite repression, you need to know about cyclic AMP and its relationship to glucose. Cyclic AMP, which is shown here and in more detail here, is a molecule used by all cells for signaling a variety of cellular processes. Cyclic AMP is made from ATP, and the synthesis pathway is shown up here in this top portion of the figure. It's something that is catalyzed. It is a chemical reaction catalyzed by the enzyme adenylyl cyclase, also known as adenylate cyclase. They're two words for the same enzyme. The important thing about that enzyme is that it is inhibited by the presence of glucose. So if there's a lot of glucose around, it will inhibit adenylyl cyclase, which will lower the overall production of cyclic AMP. In other words, high glucose will result in low cyclic AMP, or CAMP, and low glucose will result in high cyclic AMP, or CAMP. You need to remember this relationship. So we looked at a different kind of cartoon of the operon that had less detail in the previous lecture. This particular image has some more detail, and one of the things that it shows is the activator binding site. Okay, so the activator binding site is called the cap site in this particular image. Okay, and it is this purple sequence shown in here. All right, so you'll note that it is ahead of or upstream of the operator. So the cap site, cap is catabolite activator protein. Okay, so it's a DNA binding protein similar to the ones that we discussed more generally in the previous part of the lecture. We're just calling it the cap binding protein. Please note that your textbook refers to this as the CRP protein, but they're the same thing. They're synonymous. Okay? So what happens in the cell is when you have low glucose, what happens in E. coli more specifically, when you have low glucose, you have high cyclic AMP because of the impact of glucose on the synthesis of cyclic AMP that I discussed in the previous slide. So low glucose equals high AMP, which means high camp-cap complex. And when you have a high cap-camp complex, that's what's shown here, the purple blob with the little orange blob inside of it. So the little orange blob is the camp molecules, one of them is bind to the cap protein, and together they bind to the cap site. Okay? When you have that happening, you have frequent transcription because this activator protein is helping to tether this green RNA polymerase onto the promoter region. Okay? The promoter is in there. A little hard to see under all those proteins, but it's in there. Okay, and that's what RNA polymerase is binding to. So low glucose, high camp equals high camp-cap complex equals efficient functioning of RNA polymerase, therefore frequent transcription. In contrast, if you have high glucose, you have low camp, which means you will have low camp-cap complex. And that means that what the scenario shown in this figure will not happen. There will be no purple and orange cap-camp complex attached to the cap catabolite activator protein binding site, the purple site shown there. Therefore, you won't have efficient attachment of RNA polymerase to the promoter, and the result will be infrequent or low levels of transcription. This makes a lot of sense energetically, because this means that the level of glucose can control the level of expression of the lac operon. And you might wonder how those are related. But the key here is to remember that glucose is a much better source of energy than lactose. So if E. coli has to choose, you need to have a biochemical mechanism embedded in the cell that makes it choose glucose over lactose if both are present. When might both be present? Well, in your gut, in your lower intestine, right after you had a big bowl of ice cream, which would be rich in both lactose sugar and glucose sugar. So in that scenario, you want to have a way to force E. coli to use glucose, and those proteins are generally constitutively expressed, and you want to make it stop using lactose, and this is a way to do that. So overall, with the lac operon, there are two regulatory processes at work. There's the role of the repressor protein, which can be removed by allolactose, thereby allowing transcription of the lac operon. And there's the role of the catabolite repression system, whereby glucose levels control camp levels, which controls the interaction of the cap protein with the activator binding protein site, which is shown in blue here, that particular site, and that plays a role in the efficiency of transcription. It helps to tether RNA polymerase to the promoter region. So we have sort of a two-part control system, all of which control lactose catabolism. Let's think about the kinds of data that scientists studying this system might gather in the lab to help to show that this proposed model of regulation actually works in the cell under environmental conditions with varying levels of glucose and lactose. By having the regulation of the lac operon reliant upon two different methods of regulation, one linked to lactose levels and one linked to glucose levels, E. coli obtains the ability to choose whether it's going to eat glucose or lactose. And it drives E. coli such that it will have to choose glucose preferentially over lactose if both are present. And that's because glucose is a better source of ATP, as I mentioned a moment ago. This graph shows that choice occurring and the results of the regulatory processes happening at the lac operon. The red line here, shows cell growth, and the blue line here shows the level of the enzyme beta-galactosidase, which you get when you express the LAC-Z gene found within the lac operon. If you start by growing cells in a mixture of glucose and lactose, so at the very beginning you have glucose plus lactose present. You start by growing the cells in that mixture, as these authors did, and what you'll see is an increase in cell numbers, that's shown in the red line, and you'll see no beta-galactosidase initially. Why not? Well, because the glucose is degraded using glucose catalytic pathways, that lets the cell make ATP, but the lac operon is repressed because high glucose equals low CAMP, which means low CAMP-CAP complex at the activator site, which means very inefficient transcription, or essentially no transcription of the lac operon, which means no beta-galactosidase. That explains the flatness of this line in this region. So even though there is lactose present, and therefore the repressor protein will not be bound to the operator region of the lac operon, the RNA polymerase does not work well because of catabolite repression. But at the point in time where glucose is exhausted, in other words, the cells have consumed all of the available glucose, leaving them the choice of either not growing or choosing to grow on lactose, and of course they're always going to choose to grow. Now we have a different story. Now we have low glucose, which means high cyclic AMP, thus high CAMP-CAP complex, which will be attached to the activator binding sequence, a little blue C site shown on the previous slide. This promotes attachment and activity of RNA polymerase at the lac operon. Because there is lactose present, the repressor protein will not be bound to the operon, to the operator in the lac operon. Therefore, transcription can proceed, and it will proceed at high efficiency. You'll have a high rate of transcription. The lac operon will then be transcribed, messenger RNA will be produced, and the level of beta-galactosidase will increase. That's what we're seeing here. And we'll see cell growth continue after a brief plateau. That plateau is because the cell needs a little bit of time to do all of this work. And you'll notice that the steepness of the figure depicting relative cell density is less when growing on lactose than it is on glucose. And that's because glucose is a better way to make ATP. Glucose catabolism is a better way to make ATP than lactose catabolism. However, the lactose catabolism works as shown by the increase in the red line. So why is catabolite repression a global control network? Well, it's because CAMP levels, which are inversely proportional to glucose levels, control the expression of genes other than those directly involved in glucose catabolism. So in this story, the environmental cue is glucose level, the effector molecule is CAMP, but multiple genes are being affected. For example, the lac operon, which is an operon theoretically unrelated to glucose catabolism, and also many other genes in and out of operons. For example, in Bacillus species, the operon that encodes the genes for xylose utilization, another type of sugar, and also for histidine utilization, are both controlled by CAMP levels using similar mechanisms to the one affecting the lac operon just discussed. So this is why we have catabolite repression classified as a global control network. Let's consider an example of another global control system, the heat shock response. So the cells make a variety of proteins that are known generically as heat shock proteins, or HSPs, and these have been well studied in E. coli. These are proteins that help counteract cell damage from heat, ethanol, UV radiation, or other chemical or physical stressors. There are three major groups: HSP70, also called DNA-K, HSP60, and HSP10. These proteins function as chaperone proteins in some cases. Those are proteins that can correct misfolding of other proteins. That's a real problem when temperature increases. And many of them can also function as proteases to degrade other proteins that have been denatured due to high temperature. Transcription of heat shock proteins is mainly controlled by the alternative sigma factor, sigma 32, also called RPOH. This alternative sigma factor interacts with the promoters of genes that encode heat shock proteins, thereby controlling their expression and limiting it to circumstances under which the environment has signaled that it is necessary to transcribe those particular genes. Kind of a 911 response. So let's consider the basics of this particular model. What will happen under low temperature and what will happen under high temperature. So the top is showing low temperature and the bottom is showing high temperature. So at low or normal temperature, the protein DNA-K binds to the alternative sigma factor, sigma 32, also called RPOH. That's it right here. And when it binds to it, that bound complex allows the RPOH to be degraded by a particular protease in the cell. So essentially DNA-K binds RPOH so that it can hold the RPOH up so that the RPOH cannot get away from proteases, and that results in the degradation of RPOH, resulting in overall low levels of RPOH. If there are low levels of RPOH, then it's not available to bind to the promoter region of heat shock protein genes, and they don't get transcribed. But what happens is, as the temperature heats up, DNA-K will start to bind more and more with unfolded proteins. So over here we have just a protein, right? And as temperature increases, that protein is unfolding at high temperature. So this could be any protein. Okay? And there are a lot of proteins in cells. So generally speaking, as you increase temperature beyond a certain point, you'll have a lot of proteins that are unfolded. And DNA-K is a little bit fickle. So it loves unfolded proteins. So DNA-K, if it's given the choice between binding with RPOH or unfolded proteins, will always choose the unfolded proteins, forming this type of complex. Okay? And that leaves RPOH free to go off and bind the promoter regions of heat shock specific genes, thereby facilitating their transcription, and once they're transcribed, they're translated, and then we have heat shock proteins in the cells, which are available to do a variety of functions that the cell requires under high stress. So heat shock proteins is another example of a global control system. Why would that be? So why would that be? Why would heat shock response be a global control network or regulatory system? Because one environmental signal, heat or other stress such as UV exposure, also radiation exposure would trigger this, will result in altered expression of multiple genes that encode proteins that do a variety of functions in the cell. For example, DNA repair proteins, proteases to get rid of damaged proteins, and many others such as proteins involved in controlling the timing of cell division. So this is a global control network because one signal triggers control over a variety of different genes involved in a variety of different, seemingly unrelated functions. E. coli is, of course, the best studied organism on Earth, and we know that there are a variety of global control systems in E. coli. So we just talked about heat shock, okay, and we also talked about catabolite repression, but the processes of aerobic respiration, anaerobic respiration, nitrogen utilization, response to oxidative stress, and something called the SOS response are other systems wherein there is an environmental signal that triggers the activity of a particular regulatory protein that then controls the regulation of a variety of diverse genes. Okay, and I want to just draw your attention to catabolite repression, which is a phenomenon that you have understood now, or you should understand now in the context of the lac operon. That's just a few genes. In E. coli, catabolite repression, which acts through cyclic AMP and the CAP protein, which your book refers to as CRP, actually is a system that controls the regulation of more than 300 genes in the genome. So that really should give you a feeling for just how global or widespread that particular control system is in E. coli. All right, now I'd like you to go on to part 3, which is the third and final part of this lecture.